She's being loud. Okay, this is chapter 9 on cellular respiration. It's been a while since I made a video. Oh, there we go. This is an overview of the energy processes. So it includes photosynthesis and cellular respiration, and the two important organelles for that. So how they're connected, light energy comes in and it's captured by the chloroplast, actually captured by the chlorophyll in the chloroplast. And this is where photosynthesis happens. That makes organic molecules. So what do we mean by organic molecules? We mean glucose, or in a more complex form, starches. Plants also make cellulose. And oxygen. And that oxygen is then sent to the mitochondria, where cellular respiration happens. And a lot of times so cellular respiration, a lot of times people see the word respiration and they think breathing. And cellular respiration is really more about breaking down the foods that you eat. So these organic molecules, you're going to ingest them. Plants are going to use them then to convert into ATP. ATP is what we produce in cellular respiration. It's really, cellular respiration is about going from glucose or other complex sugars into ATP. That's what it's about. It's not really about respiration. Where do we get respiration from? Well, you are going to produce carbon dioxide as a byproduct, and ultimately that carbon dioxide is going to go into your lungs, and it's going to be expelled, and that's going to give back to the plants. So that's where we get it. But this is actually what's happening at the cellular respiration in your mitochondria. We're converting glucose into energy, into ATP. This is cellular energy. It's kind of like how when you plug in a hairdryer, you don't plug it into a piece of coal because that's not useful. Glucose isn't really useful to your cells. It has to be converted first before it can be used. In this process, we also release some heat energy. So think about your body heat. You're releasing energy as your cells, or you're releasing heat as your cells burn off the food you eat or burn your energy. So then carbon dioxide is released as a byproduct of cellular respiration, and also water is. The carbon dioxide is given back to the plants. The plants will use carbon dioxide plus sunlight energy plus water, and they're going to convert that to glucose and complex starches. They're going to use the oxygen that was provided in the atmosphere, and they're then going to give that into the mitochondria. The mitochondria is going to convert the organic molecules into ATP, and it's going to give off carbon dioxide, which is going to go back into the plants, and then the plants are going to give oxygen, and it just goes in a continuous cycle. So a little bit about chemistry and catabolic pathways. Catabolism or catabolic pathways release stored energy by breaking down complex molecules. This kind of goes back to chapter 8 when it talks about energy. Enzymes catalyze reactions and release energy. Some energy is used for work, and the rest dissipates as heat. Remember that last picture of releasing heat, which I can't get to. Oh, that's the wrong direction. Going the wrong direction. Nope, still going the wrong direction. Here's that picture. So remember this heat here. Well, that's part of any chemical reaction, is dissipating some heat. Fermentation is a partial degradation of sugars that occurs without oxygen. We're going to be doing some in a lab on fermentation. Fermentation is usually things that yeast do or other prokaryotic cells. I'm having trouble with my board today. Or other prokaryotic cells. Things that don't have a mitochondria, they're going to do partial respiration, and that's going to release a little bit of energy, but not as much. Aerobic respiration is what most eukaryotic organisms do. Oxygen has to be consumed to break down the sugars. it's more efficient than fermentation. So more complex organisms can do that. Anaerobic respiration is used by some prokaryotic organisms, and they use things other than oxygen, and they do something very similar to harvest their energy. Again, it's not as efficient as aerobic. Technically, cellular respiration can refer to aerobic or anaerobic. So when we say cellular respiration, we could be referring to either of them. But when we discuss it in class, we're really focusing on aerobic respiration. We'll do a little bit with fermentation too. Organic compounds plus oxygen yield carbon dioxide plus water and energy. So here's the equation for cellular respiration, the overall equation. Cellular respiration is actually a series of chemical reactions, but this is the overall. This is glucose and oxygen yields carbon dioxide, water, and this is cellular energy. Notice the numbers in front of them. That's important in balancing. If you learned about in chemistry, you have to make sure that the number of atoms that go in are the same number that comes out, because matter is neither created nor destroyed. When we talk about organic compounds, we are usually referring to glucose, a simple sugar of carbohydrates. Although proteins and lipids can also be broken down through cellular respiration, the fuel cells most often used is glucose. This is why if you are an athlete, you might carb load, and that's because that's the best source of immediate energy. We don't really start breaking down proteins and lipids until we don't have any carbs left. So this is why diets work, because you can start breaking down fats. And then if you're really, really starving, your body might even start to break down proteins to get energy. Cellular respiration is exergonic. That means it occurs spontaneously. It has a negative delta G. If you remember this from chapter 8 about free energy and enzymes, you're also going to be watching video 12 on life requiring free energies that will review that for you. Apparently in chemistry, you guys don't learn about redox reactions. So I'm going to try to explain this as easily as possible without too much depth, because you don't really need to understand it too much, but just a general idea. The relocation of electrons releases energy from organic compounds and is used to synthesize ATP. So a redox reaction is also called an oxidation-reduction reaction. The loss of electrons from one substance is oxidation. So if something becomes more positive or has a higher proton amount, it's because it's losing electrons. The addition of electrons to another substance is called reduction, and that's going to cause it to become more of a negative value. And the reason is because we're adding more electrons, and that makes the substance more negative, hence why we have the negative behind it. This is an example. So in this reaction, sodium is becoming oxidized, so it's actually losing electrons and becoming more positive, and then chlorine is going to gain electrons and become more negative. This is really not my field. This is more chemistry. I'm giving it to you kind of as short as possible. In this equation, glucose becomes oxidized, and oxygen becomes reduced to water. In cellular respiration, glucose is oxidized and carbon dioxide is reduced. If energy were released all at once, it would not be as efficient, and therefore cellular respiration releases energy from glucose in steps catalyzed by enzymes. So even though I showed you the whole equation for cellular respiration, it actually happens in multiple simple steps. We're actually going to do an activity in class where we look at the Krebs cycle in particular, and we're going to look at some of those specific steps of how that process happens. During this process, at key steps, electrons are stripped from glucose. NAD+ is an important electron carrier in cellular respiration, which cycles between NAD+, which is the oxidized form, and NADH, which is reduced. And so this is important in electrons being transported during the process. We want to make sure we have a lot of electrons available at the end. So NAD+ is often going to be reduced. Most electrons in cellular respiration follow the path glucose, NADH, electron transport chain. So like we said, it's an electron carrier, so it's going to put out a lot of electrons, and those are going to be important later on in the electron transport chain. And then oxygen, with oxygen being the final electron acceptor. So all of these electrons that we're gaining by NAD plus being reduced, we're going to put into oxygen. Remember, oxygen is highly electronegative. The stages of cellular respiration preview, summarized, is glycolysis. This happens in the cytoplasm, and we don't need any oxygen for this part. It results in pyruvate. Okay, so what do you need to know ultimately when we look at the equations? This is what you're going to need to know. Okay? The steps in there aren't quite as important as the main what starts it and what ends it. So here we start with glycolysis. We're going to start with your glucose and we're going to end in pyruvate. Then pyruvate is going to go through the citric acid cycle and result in acetyl-CoA, so coenzyme. Oxidative phosphorylation is the electron transport chain and chemiosmosis in the inner mitochondrial membrane. Electrons are going to be passed down and ATP will be synthesized. This is where we're making all of that ATP, is through chemiosmosis. We're going to talk about that. And you often want to think osmosis and water, and it really has absolutely nothing to do with water. So this kind of summarizes it as well. So glycolysis happening on the cytoplasm. Glycolysis means sugar splitting. So we're going to break down sugars. It occurs with or without oxygen. It doesn't really matter if it's there. It uses energy to make it happen, but pays off with more energy. It takes glucose and creates 2 pyruvates and 2 ATPs. That's what you need to know. Net. Remember, we actually use 2 ATP into the system, so it's really a total of 4, but it's 2 because you have to subtract the 2 you put into it. And we also use 2 NADHs. So here's glucose glycolysis summarized. And then we go on to the next part. Now we're moving into the mitochondria, and we're going to take pyruvate and turn it into acetyl-CoA. If oxygen is present, important, pyruvate will move into the mitochondria. Pyruvate is oxidized. Remember, this means it loses electrons, and it's going to turn into acetyl-CoA, which will then enter into the Krebs cycle. So here we have pyruvate, and that's going to turn into acetyl-CoA. And through many steps, acetyl-CoA will go into the Krebs cycle and produce 2 ATP, 6 NADH. So remember, we're donating more and more of these to the end result, 4 carbon dioxides, and 2 FADH2s. This is the Krebs cycle. It's also sometimes called the citric acid cycle. And you do not have to know all of these steps here. You just need to understand that one thing turns into another, which turns into another, which turns into another, which turns into another. And we'll do some of this, and you'll actually create some of these molecules. But ultimately, the big idea is we're going from acetyl-CoA or acetyl-CoA going in here, and we're going to produce a little bit of energy along the way. We're going to produce some NADHs that will then be sent onto the electron transport chain. And here's kind of a bigger picture of it here. Pyruvate, remember, into acetyl-CoA, and then that's going to go through the citric acid cycle. So here's the citric acid cycle. This shows you here where you're producing those NADHs that will eventually go on to that electron transport chain. And where are you producing energy along the way? A little bit of energy here. This is where you get your big bang buck of energy, and then this is your total energy. So the electron transport chain, we're going to use the NADH from the previous reactions, and FADH2 are going to transfer your electrons to the electron transport chain, and then those are going to move down the chain, losing energy in several energy-releasing steps. And finally, the electrons are going to be passed to oxygen. Remember, that's the final electron acceptor, and it's going to reduce it to water. So here's a picture of those steps along the way of how we're going to ultimately reduce oxygen to water, and we're going to use the NADH, and we're going to do all these complex things, and we're going to turn Oxygen is going to accept those electrons, and we're going to reduce it to water. Chemiosmosis is probably one of the most important steps to understand about cellular respiration. During certain steps, hydrogen ions are moved to the intermembrane space of the mitochondria. And they're going to store energy as a protein motive force. This is interesting. We'll talk about it. Or a hydrogen plus gradient, so a proton gradient. When the protons move through the ATP synthase, its passage drives the phosphorylation, adding phosphates, that's what phosphorylation means, is adding phosphates, to ADP to form ATP in the mitochondrial matrix. This is what we call chemiosmosis, so it has nothing to do with water. It's chemical formation of ATP. So here's a picture of it. So we're going to put all these hydrogen ions, we're just going to gather up a whole bunch of hydrogen ions and create a high amount of hydrogen ions up in the intermembrane space. That's going to be happening all along the electron transport chain. Ultimately, what that ends up with is you have a lot of hydrogen ions up here, and those are actually going to power this ATP synthase. It is a membrane rotor, I guess is a good word for it. It's actually a physical machine almost that is powered by these hydrogen protons. They're going to power it, and that's going to take ADP, which is already in your cells, adenosine diphosphate, so 2 phosphates, and it's going to add phosphates and make ATP. That's what chemiosmosis is. It is using this proton gradient, or all these extra protons, to power a rotor that will turn ADP into ATP. That's what's happening there. It looks kind of like this, or at least the animation looks like that. So you've got all of these hydrogen atoms up here, ions, and those are going to power this, and that's going to turn ADP into ATP. It actually is a physical internal route, like it's physically powered by the chemical proton gradient here. Accounting for ATP production. The net is about 28 ATP from the electron transport chain alone. So it by far is producing the most energy. Add in the 2 from before, and the net total is about 32. It can fluctuate from 30 to 36, but it's actually very efficient. Electrons follow the path of glucose into NADH down to the electron transport chain, and then through that protein motive force. That protein motive force is this rotor. And then they're transformed into ATP. About 34% of energy that is stored in glucose is transferred to ATP. Much energy is dissipated as heat. Okay, so we do lose a lot of heat here. It's efficient when you compare it to anaerobic respiration or fermentation, but ultimately it's not extremely efficient. Read about hibernating animals and how cellular respiration is different. They don't produce as much ATP, but they generate heat. So check out page 177 of your textbook and jot down some notes about how cellular respiration is different in hibernating animals, because they don't produce very much ATP, but they generate a lot of heat, and that keeps them going. So this is an overview of the whole thing. So we have glycolysis first. You're going from glucose to pyruvate. Then you have pyruvate oxidation, where you're going from pyruvate to acetyl-CoA. The acetyl-CoA is going to go through the citric acid cycle, and then that's going to send all of these electrons to the electron transport chain, which is going to generate the majority of your ATP. And how are we doing that? Because always in here, we're also releasing a bunch of hydrogen atoms, and those are going to power the chemiosmosis, the rotor that's going to turn ADP into ATP. How is fermentation a little bit different? The difference between fermentation and anaerobic respiration is that the electron transport chain is used in anaerobic respiration, but not in fermentation. That's how those two are different. A lot of times we actually use fermentation and anaerobic interchangeably in terms, but there is a slight difference in it. Anaerobic takes place in some prokaryotes, so bacteria, and uses the electron transport chain, but does not use oxygen as the final electron acceptor. Instead, it uses other electronegative substances, and it's not important to know what those are. I'm not even sure I knew that. Fermentation is a way of harvesting chemical energy without using either an oxygen or the electron transport chain. Remember, glycolysis produces 2 ATP regardless of oxygen and occurs in fermentation too. Fermentation is actually an extension of glycolysis. And this is something you would see like yeast do. We're going to look at yeast in the classroom. So this is just showing the difference here. So in fermentation, we don't have any oxygen, and we're going to produce actually byproducts of ethanol or lactate. And then over here, we've got oxygen present, and we're going to go into the mitochondria. We're going to go through the whole process, and we're going to produce more ATP than here. Alcoholic fermentation is when we go from pyruvate is converted to ethanol. So think about making wine or beer. Okay, those are all things that use yeast. Or wine actually uses grapes, they ferment to produce alcohol. Lactic acid fermentation makes lactate as an end product, which releases carbon dioxide. This actually occurs in human muscles when oxygen is scarce, like during strenuous exercise, so when you're lifting weights. Cells will switch from aerobic to fermentation when they run out of oxygen. Lactate was previously thought to cause that muscle fatigue or that pain that you got when you were exercising very vigorously, but recent studies suggest that instead that's actually increased levels of potassium. Maybe you would have blamed for that. When I was in school, we were taught that it was lactate. And so this shows the difference between alcohol and lactic acid fermentation. Really, the difference here that you need to understand is ethanol is produced versus lactate being produced. Comparing fermentation and anaerobic respiration. Both of them use glycolysis, but they're different in what their final electron acceptor is and whether or not they use the electron transport chain. Respiration yields more ATP than fermentation, but still, anaerobic is going to yield less ATP than aerobic. What's the evolutionary significance of glycolysis? Glycolysis occurs in nearly all organisms and is thought to have evolved in early prokaryotes before there was oxygen in the atmosphere. I think that's very interesting to think about that. Why do they think that? Well, glycolysis occurs in all organisms, so clearly that had to come before aerobic respiration, which requires oxygen and does not occur in all organisms. So we think that after oxygen was available, more efficient ways of doing respiration evolved. Because remember that prokaryotes that do anaerobic without oxygen, they're only going to produce about 2 ATP, whereas aerobic is going to produce, 30 to 36 ATP. We don't usually eat glucose, but attain it through fats, proteins, and sucrose, and other disaccharides like carbohydrates, complex carbs. Carbohydrates are hydrolyzed to glucose before entering into cellular respiration. So understanding that what you eat is going to be broken down into glucose before you can do cellular respiration. Proteins must be broken down into amino acids, which are used to build new proteins, which are intermediate of glycolysis in the Krebs cycle. I'm going to show you a diagram here in a minute. Fats are converted into an intermediate in glycolysis as well. Figure 9.19 talks about where different things that we eat enter the process. So here you go. So this is the main source of energy. Going to go right into sugars and right into the process. Fats are going to come in right about here, so in the middle of the glycolysis stage, and then proteins are going to come in any of these stages in the middle of glycolysis, where you're converting to acetyl-CoA or even into the citric acid cycle. This is kind of a last resort of burning energy if there are no carbohydrates or fats available. Glycolysis in the Krebs cycle connects to other metabolic pathways. Cellular respiration is controlled by enzymes at key points in glycolysis and the Krebs cycle. So remember, enzymes are really important because they help turn one molecule into another. They catalyze any of those reactions. We learned about those, remember, before in chapter 8. And so this just gives you an example of phosphofructokinase. This is an enzyme that's catalyzing the process of fructose 6-phosphate into fructose 1,6-biphosphate. You do not need to know those chemical compounds, but it's just showing you that this enzyme is important in making this happen. Well, what happened is it actually inhibits the process later on that will then help drive the rest of the process. AMP is adenosine monophosphate, which actually stimulates the activity of that enzyme. And when we produce ATP, that's going to then inhibit this enzyme because we are already producing it. If we need to produce more, then it's going to be stimulated. And that's it. We are going to talk a little bit about this in class. We're going to do a little bit of modeling. Bozeman has some great videos on this as well to help you.